Okay, welcome back. In this video, we will continue our lecture series on pharmacology for the health professional. In this video, we will discuss chapter number 2, drug design, testing, manufacturing, and marketing. Learning objectives for this chapter: name several ways in which drugs are discovered or created, describe how computers facilitate drug design, differentiate between chemical, generic, and trade or brand names of a drug, list at least 5 things that the trade names of drugs might tell you about those drugs, describe the process of a clinical drug trial, describe how inert ingredients might affect the bioavailability of a drug, describe how direct marketing of prescription drugs has affected consumers and drug costs, give 4 reasons why a drug might be withdrawn or recalled, and lastly, define the keywords and phrases for this chapter. First, a quick introduction. The development and the testing, manufacturing, and the eventual marketing of a drug is a very time-consuming, very expensive process. Now, a drug company may evaluate thousands of different chemicals before finding one that goes all the way through the process of all the phases of testing and then is finally approved by the FDA to be released and marketed. Drug discovery and creation. Now, drugs are discovered or created in several ways. One of those is using ancient sources. Another way would be the discovery of a new drug. Get this by deriving a new drug from the molecular manipulation of a currently existing drug and creating a brand new one, genetic manipulation, and also gene therapy. The first topic we mentioned, ancient sources. Now, many drugs that are still in use today were originally derived from either plants or animals or mineral sources hundreds or even thousands of years ago. Now, a totally new chemical can be discovered in the environment or from plants or from animals or the ocean or the soil. For example, Taxol, which is a chemotherapy drug. This is taken from the needles of the Pacific yew tree. The anti-tuberculosis drug, streptomycin, is from the stomach of sick chickens. Another example would be the fungus from which cephalosporin antibiotics are derived. You can also have a totally new chemical be derived from the molecular manipulation of a currently used drug. And by doing so, you are creating an isomer. And by doing so, you're creating dextrorotatory drugs and levorotatory drugs. And both of those terms relate to the property of rotating the plane of polarized light, either to the right or to the left. Dextro would be toward the right, and levo means toward the left. Sometimes you'll see just the capital D used, or the capital L used. In this example, we have the first benzodiazepine anti-anxiety drug that's created. That's here on the left. Now, starting from this currently existing drug, by manipulating the molecular structure, you're able to produce diazepam. And both of these drugs are currently in use to treat neurosis and anxiety. But diazepam was created starting with this as a precursor. Now, previously, designing a new drug by changing the molecular structure of a currently existing drug was a very time-consuming, very slow process, and most of it was just trial and error, using either intuition or molecular models, ball and stick models of compounds. But now, with computers, it is much easier to complete this process. You can use a computer to display the molecular structure of any drug, so you're able to identify unsuccessful chemicals before you invest large amounts of money, large amounts of time to see whether or not they work. Now, even with very slight molecular changes, the original drug may be significantly changed. Changing a double bond to a single bond, adding a methyl group here versus here can mean a totally different outcome of a drug. And that leads you to molecular pharmacology. This is the study of chemical structures of drugs and their effects at the molecular level within the cell. So not just the drugs themselves, but how they impact the cell at the molecular level. You can also generate a new chemical through genetic manipulation. And by doing so, you are creating recombinant DNA, or rDNA. You are recombining DNA to get a new structure, a new compound. And it's done so by gene splicing, or genetic engineering, or in vitro procedures, gene cloning, and the creation of human insulin, or humulin, for example. Another avenue that can be used to create new drugs is gene therapy. This is largely based on the results of the Human Genome Project, where the entire genetic information of a person was able to be decoded. So knowing what genes are there and knowing what they do can help pinpoint what is wrong with a person and how to best treat that disease or that condition. When you combine pharmacology and genetics, that leads you to a whole new area of research, such as pharmacogenetics and pharmacogenomics. Pharmacogenetics, a branch of pharmacology that deals with the genetic factors on reactions to drugs. And pharmacogenomics is a broader term, and it deals with how a person's genome, how their genes can influence their response to a drug. So pharmacogenetics is a reference to how a variation in one single gene can influence a person's response to a drug, and pharmacogenomics is how their overall genome, how all of their genes can influence a response to drugs. Okay, so by knowing all of a person's genetic information, you are able to pin down what every individual gene does. And by doing so, this is creating a CMAP, or connectivity map. So this is a catalog of gene expression data. Alright, this image, we have an example of Humulin, which is a first drug to be created with recombinant DNA technology. This is a human form of insulin. And we look on the box, you'll see rDNA origin, recombinant DNA origin. Now we'll move into drug names. Every drug has a chemical name, and the group that will describe the molecular structure is IUPAC, the International Union of Pure and Applied Chemistry. Drugs also have a generic name, and the United States Adopted Names, or USAN, determines what a drug's generic name is. You also have a brand name or a trade name. These are created by the drug company that manufactures the drug after that drug is approved by the FDA. Now these trade names are designed to be easier for physicians and patients to remember, as opposed to a very long, cumbersome chemical name. And it's also used to suggest how the drug is used. And these trade names are a trademark symbol. They are a part of the marketing of that drug. In this image, we have an example of a diuretic drug. Chemical name of this compound is listed here in the caption: 6-chloro-3,4-dihydro-2H-1,2,4-benzothiadiazine 7-sulfonamide 1,1-dioxide. So good luck getting that on a commercial. That's the chemical name for this structure. The generic name of this drug would be hydrochlorothiazide, and the trade name for this compound would be hydrodiaryl, or could also be sold under microzide. So here you have the very long chemical name, which is a detailed description of the compound. Then you have the generic name, then you have the trade name. The trade name is what you would see on advertisements and commercials. Alright, in this image we have a brief list of drug references. Now when you see a list like this, you see a drug's therapeutic use, the drug forms that it comes in, and doses. And we see names of drugs like this. The generic name always begins with a lowercase letter, and then the trade name, brand name, always begins with a capital letter. And for example, the trade name Acunev starts with a capital A. That's the trade name. That's how that would be sold and marketed to doctors and to patients. The generic of that is albuterol, identified by the lowercase a. So one's the generic name, one's the brand name. Accurate spellings of drug names are critical, especially when you deal with very long and confusing names, and there are no linguistic standards when it comes to this. So some trade names are difficult to spell because drug manufacturers aren't held to any kind of standards. And for example, the trade name drug Rhythmol is used to normalize the rhythm of the heart. But there's no H found in the trade name, even though there's a H in rhythm. So there's no linguistic standards that would include having a letter there or not. Alright, there are some tips that you should follow when designing drug names. Tip number 1, the spellings of generic drugs belonging to the same category often reflect chemical structure. However, the spelling of the drug is often different from the spelling of the disease. For example, Asmacort is used to treat asthma. Mucinex is to remove mucus from the lungs. Rhythmol, like we talked about in the last slide, is to treat abnormal heart rhythms. Tip number 2, the spelling of the trade name drug indicates what part of the body is being treated. Boniva is used to strengthen bones, so bone Boniva. Bronchaid is used in dilating the bronchioles in the lungs, so Bronchaid bronchioles. Nasal-Crom is used to treat nasal allergies. So the trade name gives you a good clue on what is being treated. Tip number 3, the spelling of the trade name simplifies the generic name, but still retaining parts of the phonetic sound. See, the trade name Cipro is a shortened form of the generic drug ciprofloxacin. Haldol is a trade name, short for haloperidol. Humulin, which we talked about earlier, that's the trade name. The generic name, human recombinant DNA insulin. Sudafed, that's a shortened form of the generic drug pseudoephedrine. Tip number 4, the spelling of the trade drug name indicates the ingredients or the source of the drug. For example, Ferenzol, this is iron. The chemical symbol for iron is Fe in solution. So iron in solution, Ferenzol. Chloride chloride, potassium chloride. K is the chemical symbol for potassium, Cl for chloride. Premarin, pregnant mare's urine. Pre for pregnant, mare is reference to the mare, and then in, urine. Tip number 5, the spelling of the trade name indicates the therapeutic effect of the drug. The Elamite eliminates mites, or scabies. Glucotrol controls levels of glucose. Lipitor decreases levels of blood lipids. Restoril restores sleep to treat insomnia. So you are restoring sleep. Tip number 6, the spelling of the trade name indicates how often the drug is to be taken. Lithobid is a lithium drug given twice a day. Litho for lithium, Then BID, Latin for twice a day. Nitrobid, nitroglycerin given twice a day. Nitro for nitroglycerin. And again, BID means twice a day. Tip number 7, the spelling of the trade name indicates the duration of the drug's effect. See, Cardizem LA, long-acting drug for hypertension. That's what the LA stands for. Pronestal SR, the SR stands for sustained release drug. Then slow FE, FE is a reference to iron, the chemical symbol for iron. And then slow, pretty straightforward, means a slow release. So that would be a slow release iron supplement drug. Tip number 8, the spelling of the trade name indicates the strength of the drug. Bactrim DS, the DS stands for double strength. Cortisone 5, that 5 is a reference to the percentage of hydrocortisone. So it is a 0.5% hydrocortisone anti-inflammatory ointment. Tip number 9, the spelling of the trade name indicates the route of administration. Transderm scope. Transderm is a reference to transdermal skin patch. Tip number 10, the spelling of the trade name indicates the amount of the particular active ingredient. We have 2 examples here. Both involve Tylenol. Tylenol with codeine number 2, that contains 15 milligrams of codeine. Tylenol with codeine number 3 contains 30 milligrams of codeine. Tip number 11, the spelling of the trade name reflects the drug company's identity. ED TUS HC cough syrup is manufactured by the Edwards Drug Company, ED for Edwards. See, the drug Witensin for high blood pressure are manufactured by the Wyeth Ernst Drug Company. Alright, next we'll talk about the testing of new drugs. See, all drugs must be thoroughly tested by the drug company before it can be marketed according to FDA guidelines. Now the testing is done to determine the drug's effectiveness and also its safety. There are many different methods of testing. One of those is in vitro testing. The phrase in vitro is Latin, which means in glass. This is done in test tubes. This is where you have chemical analysis. In vivo testing. In vivo is Latin for in living. So this is where you have testing done on living specimens like animals or humans. When it comes to animal testing, this always precedes testing on humans. During this process, the drug will be evaluated for side effects, toxic effects, addictions, the development of cancerous tumors, any fetal deformities, and also for pharmacodynamics. Now, pharmacodynamics is the study of the effects of drugs, the actual mechanisms of their actions. And that would include various aspects of how a drug would work, including frequency distribution, a drug's half-life, and that's the time it would take for a substance to lose half of its pharmacologic or radiologic effect. Median effect dose, or the ED50. So this is the dosage that is required to receive 50% of the desired response. That's why it's ED50. A median toxicity dose, or TD50. That's a dose that's required to get 50% of the population who takes that drug reporting a specific toxic effect. And then the TI, the therapeutic index, is also known as the safety window or the therapeutic window. And this concept is the relationship between the toxic and the therapeutic doses of a drug. And some drugs are defined as having a narrow therapeutic index. And what that means is very small differences in dose could lead to some serious adverse effects with that drug. Right, after we have the completion of the animal studies, the drug company will then submit an IND, an investigational new drug application. Now this application will have information about the completed animal trials. It will supply information that the drug does not have a risk to humans, and it will include information about the drug such as the manufacturing processes and the chemistry of the drug. Now when it comes to testing drugs on humans, there are multiple phases. These are called clinical trials. The first one, phase 1, this will be done on anywhere between 10 to 100 healthy volunteers, and it is mandatory that they have informed consent. And this phase of the trials, you're looking to evaluate any possible side effects to establish the final and the correct dosage for what you're trying to treat. And you also are testing for pharmacokinetics. How well is this drug being absorbed and distributed and metabolized throughout the body? And this phase of the trials will take between 1 to 1.5 years. After phase 1, we move on to phase 2 of the clinical trials. This will take anywhere between 50 to 500 patients who currently have the disease that you're trying to treat. And this drug is now given on an experimental basis. And this phase is used to determine the therapeutic effect of your drug. And this phase takes about 2 years. Now what's commonly used in clinical trials is a placebo. And a placebo is a drug that exerts no pharmacological effect and no therapeutic effect and has no side effects when administered. And the word placebo actually is from Latin, which means I will please. It doesn't help you, it doesn't harm you, and it has no side effects. Now placebos are used in double-blind research studies, and that means that neither the researcher dispensing these drugs nor the patient knows whether or not the drug they are taking is a real drug being tested or a placebo. And that way, by no one knowing is it the real drug or is it just some sugar pill, the results aren't going to be tainted. Now, placebos are commonly sugar pills or injections of sterile saline solution. Now, while it is physiological impossible for a placebo to have any kind of pharmacological effect, patients will often report a decrease in certain types of symptoms and can experience side effects when given a placebo. Now, these effects are quite real and demonstrate that in some situations, the powers of suggestion can produce changes within the body that closely mimic pharmacological effect of an actual drug. Now we move on to phase 3 of the clinical trials. This will involve several hundred or several thousand ill patients. And at this stage, the drug will be administered the same way it will be used on the market. Frequency, dosage, route of administration, everything would be exactly the same as if it were already approved to be sold. Now, the performance is compared with other drugs that are currently on the market, is where you'll have double-blind studies with placebos performed, and this phase will take about 3 years. And then when it comes to testing on children, this is the phase where you get the standardized pediatric doses, because obviously the dose that you would give an adult will vary from what you would give a child, and the manufacturer of these drugs will receive a 6-month extension on a standard 17-year patent. Alright, upon completion of phase 3, the drug company will submit all the documentation to the FDA in a new drug application, NDA. At this point, all they can do is wait for a final decision from the FDA, and the drug will either be approved or denied. And only about 20% of NDAs receive a final approval from the FDA for marketing. So it's very likely that you'll get all the way through the end of phase 3, complete all this research, invest all this time, all this money, and your drug will still get denied. Once the FDA approves of the drug, the ingredients, the dosage, the manufacturing process, labeling, and packaging cannot change. Now, the drug company can conduct further clinical trials to expand the use of that drug. Did you know the data that's collected for just one patient in just one clinical trial can exceed 100 pages of documentation, and the total documentation for all aspects of a drug testing process can exceed 100,000 pages. The ulcer drug Tagamet is a good case in point. After 4 years of testing, the SmithKline Company had accumulated a stack of documents that stood 17 feet high that had to be taken to the FDA in a truck. So that's how detailed this information has to be. Alright, now move on to drug manufacturing. Now, in the past, pharmacists would commonly mix drugs. And today, of course, the manufacturing process is a lot more regulated, especially when it comes to drug quality, sanitation, and then packaging of drugs. Now, generic drugs and trade name drugs, regardless of who manufactures them, must have the same dose strength, they must contain the same active ingredients, and they both must be administered in the same way. Now, manufacturing of generic drugs and trade name drugs, regardless of who manufactures them, does not apply to the bioavailability of the drugs, the inert ingredients, the preservatives, the antioxidants, and the buffers used. The process that includes securing drugs in appropriate containers also, such as adding a packet of desiccant, which is a drying agent for drugs. Also tightly sealing the top to prevent tampering, and also replacing individual drugs in blister packs. This also includes the expiration date on every drug label, and all drugs must have a NDC number, which is a unique identifier number for the National Drug Code. Alright, this image we have examples of protective packaging. All these are individual blister packs, so the only way you can access these individual drugs is to poke it through that blister pack. So this is to help to prevent tampering. In this image we have an example of a National Drug Code. Every prescription drug has a NDC code, and this will include the drug company that manufactures it, the drug strength or dosage, and the package size and the type. Now, the NDC may also appear on the prescription bottle or other printed labels generated by your pharmacy. So, for example, this is how it would appear for this drug, amiodarone, which is a drug used to treat heart arrhythmias. So you have the drug name here, you have the dosage, 200 milligrams, the quantity, 40 tablets, the actual NDC number for this drug, then the manufacturer, Eon Labs. Alright, next we'll talk about drug marketing. The regulation of drug marketing will vary depending on whether or not the drug is a prescription drug or if it's over-the-counter drug. The drugs that are over-the-counter are regulated by the FTC, Federal Trade Commission. Advertising of prescription drugs is regulated by the FDA. Now, previously, drug companies would promote prescription drugs by applying directly to physicians by using sales representatives, offering free samples, offering literature and videos, and this is still the most prevalent form of prescription drug advertising. That's how it worked in the past, but now marketing works by DTC, direct to consumer. This is done by magazine ads, television ads, radio ads, and depending on where you see these ads, they'll be more focused on that target audience. For example, drugs that advertise in men's magazines may focus more on erectile dysfunction drugs or drugs that would treat an enlarged prostate. Drugs that would advertise in women's magazines would focus on menopause drugs or infertility or osteoporosis or birth control. So you're going after a much more targeted audience based on the TV show you're advertising on or the magazine that you're advertising in. And with the direct-to-consumer advertising, we've gotten familiar with this phrase: ask your doctor if this drug is right for you. Now this phrase has created the consumer-driven shift when it comes to marketing. So now you are having patients proactively asking about certain prescription drugs. And they may not even need that drug, but they are asking for it. So this pressures the physician to prescribe drugs that are unnecessary or drugs that are more expensive than what that patient actually needs. All right, now we'll talk about some key points in the drug marketing timeline. In 1981, the first direct-to-consumer print advertisement appears for an analgesic drug. 1985, the FDA rules that drug ads marketed to consumers must include risk information. That's why you hear every possible side effect that might ever happen, in a TV commercial or in a print advertisement. 1997, the FDA allows drug manufacturers to direct market their ads to consumers via television. 2001, 100 prescription drugs are regularly marketed directly to consumers. So now we have slogans are becoming well known even for prescription drugs. The little purple pill for Nexium is an advertising slogan that people can recognize because it is a product. 2004, Cialis, a drug that treats erectile dysfunction, advertised during the Super Bowl. Alright, now we'll talk about drug patents. Now, the trade name of a drug is registered with the U.S. Patent Office and is a registered trademark. That's why you see the letters TM above the trade name. Once this is under patent, the drug company has the right to advertise and to market that product. And this lasts for a 17-year period. And this includes the testing period before the approval. And this law from 1984 dates back 5 years to that. Or once that trade name patent expires, competitors can't enter the market. And the drug's original name can only be used by the original company. And if a generic is manufactured, it is listed under different trade names. Alright, drug withdrawals and recalls. Now, having a drug approved is no guarantee that it will stay on the market forever. Once a drug is approved, there has to be post-marketing surveillance. So this is where you have drug companies and the FDA continue to monitor that drug after it has been approved to make sure that there are no other side effects that were not known at the time of its initial approval. So it may be weeks, months, or even years after the approval process has finished that some adverse effects may become apparent. So this is where you have health professional reports on whether or not a drug is effective or not, and also the FDA's MedWatch. So a drug is continually evaluated for effectiveness even after its approval, and especially those drugs that include death. For example, antidepressant drugs had to change their warnings to include the risk of suicide in 18 to 24-year-olds. That wasn't in the initial approval process because it wasn't known until after the drug had been approved. So you have to go back and reissue new, clearer warnings based on the new information that is available. Now, a drug can be recalled when some steps have happened. For one, the drug does not contain the correct amount of the active ingredient, it doesn't remain stable until its expiration date, or the drug becomes contaminated with particulate matter during the manufacturing process. For any one of these three reasons, the manufacturing process can be stopped and every drug recalled that is currently on the market. And it is the responsibility of the drug manufacturer to notify hospitals and doctors and pharmacies of a drug recall. Alright, that brings us to the end of chapter number 2 in our video series. We will continue our video series with our next video on chapter number 3, drug forms.